My name is Colton Blake, and I'm doing my presentation on a genus of parasites known as Cryptosporidium, which includes a lot of species. Now, this is a genus of single-celled microscopic parasites, one of the most common parasites contracted by a wide range of animals, including humans. This parasite is usually detectable in feces, but it can be challenging to find due to its incredibly small size, being just a handful of micrometers. I actually volunteer in a diagnostics lab where I'm getting a lot of experience in extracurricular learning, and I can say from firsthand experience that most of the parasites we find under the microscope are much larger than 10, 20, or even 50 micrometers. So 1 to 3 micrometers is incredibly small. It's usually about the size of bacteria, while most eukaryotic cells are much larger than 10 micrometers. So in order to detect this organism, a common test is the immunofluorescent assay, seen on the top left here, where these antibodies with a fluorescent protein stick to the surface proteins of Cryptosporidium, creating this green color here when viewed under the right microscope. But of course, there's other ways to see the parasite as well, such as in the acid-fast test, seen on the bottom left, where essentially the parasite is dyed purple and nothing in the background will absorb the color, or maybe a few other organisms, but that's a long shot. And then you get this image of these purple organisms sticking out against a clear background. Cryptosporidium belongs to the domain eukaryotes. This includes organisms with a bit larger cells. Of course, this is an exception. It also includes organisms whose cells have nuclei and chromosomes as opposed to no nucleus and circular DNA. In addition to that, most eukaryotes will have a little bit more complexity, and they will have membrane-bound organelles, which Cryptosporidium obviously has. It's also included in the kingdom of protozoa. Now, protozoans are eukaryotes that aren't plants, they're not animals, and they're not fungi. So this obviously includes a lot of unicellular organisms such as Cryptosporidium. Now, the phylum Apicomplexa includes all of these parasites with an apical complex, allowing them to enter a host cell and invade in that sort of way. And the class, order, family, and genus are actually highly specific to Cryptosporidium. The order Eucocitiodora actually includes only a handful of other parasites, such as Eimeria, which pretty much has an identical life cycle to Cryptosporidium. And this parasite is responsible for more than half of all waterborne disease outbreaks in the United States. And that life cycle that I was talking about over here is essentially where you consume an oocyst from the environment, and that oocyst is ingested into the host, and then the parasite is released, can enter a cell in the GI tract usually, and reproduce asexually, get thousands upon thousands of individuals, and then eventually invade again and again and again, wreaking havoc and damaging the tissue of the GI tract, and then eventually entering another cell and reproducing sexually to produce that oocyst, which can go back into the environment to be immediately infective and a bit hardy, to be found usually in water sources. So this apicomplexan parasite uses its morphology, just like other apicomplexans, to invade host cells by injecting effector proteins from this morphological feature called an apical complex, which can actually enter the host cell and inject these effector proteins. Now, what is an effector protein? Well, one 2021 study in the journal Cell Host and Microbe finds that one specific effector protein, ROP1, is injected by C. parvum into the host cell and allows for an infection to take place. Now, this happens because ROP1 disrupts host protein LMO7 by binding to one of its domains, inhibiting its interaction with other proteins. Now, LMO7 is a cytoskeleton modulator and regulation protein, and this allows for the rapid polymerization of actin when regulation no longer takes place. So the effector protein, ROP1, enters the host, disrupts LMO7 from binding with other proteins, and then that disrupts the regulation from taking place, and thus this rapid polymerization of actin will pull the parasite into the host cell at the infection point. And the publication describes the effects down here in this direct quote that I pulled. ROP1 thus may disrupt LMO7-dependent processes by blocking its interaction with its natural partners that take place in this interaction of regulating the cytoskeleton on the outside of the cell. Let's talk about where this all takes place. The parasite resides in a particular vacuole just under the cell membrane of the host enterocyte cells. Now, these enterocyte cells are the cells that line the inside of the small intestine in the gut. So this vacuole that the parasite resides in extends under the plasma membrane, but still protrudes out into the lumen of the small intestine along the epithelium. And so you can see on the left side of the image here, those microvilli, that is where this vacuole is going to be found. And this allows the parasite to rapidly enter, reproduce both sexually and asexually, and eventually burst out into the small intestine, wreaking havoc on host tissues and damaging the host and spreading throughout the gastrointestinal tract. And this causes fever, weight loss, dehydration from the body trying to flush the parasite out, and obviously damage to the tissues. So, in short, simple terms, the parasite enters these host cells on the inside of the small intestine by hijacking their defenses and structures, by interrupting the processes using this protein to break in and use the host resources and machinery to multiply inside of the cells, eventually bursting out and spreading throughout the GI tract to wreak more havoc. And of course, this causes some damage. So you can see down here in the life cycle, we'll start from trophozoite. That's just a more mobile, infectious kind of form of cryptosporidium here. So it enters our host cell in the epithelium and then multiplies very rapidly asexually, and then it'll burst out, and then it might happen again. And then down here where it says microgametocyte, it'll actually go into another host cell, invade, and start reproducing sexually. That's when you get a little bit more variation, and then they'll of course form the oocyst, which is shed out into the environment for another animal to eat and for the cycle to repeat itself. How is this treated? As I mentioned earlier, there's a lot of dehydration, so intensive hydration, supportive care in immunocompromised individuals, such as the administration of drugs which slow down the contractions and movement in the gastrointestinal tract, and this will actually reduce the loss of fluids. And these drugs that slow down these contractions are actually classified as opioids because they do work by blocking receptors in the nervous system between the muscles along the GI tract and the nerve endings themselves, and that causes the muscles not to contract. So in severe cases, drugs like nitazoxanide can be used to directly target the parasite. This kills the parasites. We'll talk about how that works on the next slide. Okay, so nitazoxanide kills cryptosporidium. How does it do it? Well, cryptosporidium uses anaerobic respiration rather than our version of aerobic respiration. But however, both of these forms of respiration to create energy use electron transfer chains. Now, in cryptosporidium, this electron transfer chain requires acetyl coenzyme A, which is a chemical derived from pyruvate. And this is done by an enzyme called pyruvate ferredoxin oxoreductase, or P4, which is responsible for converting pyruvate into acetyl coenzyme A, which is necessary to begin the electron transfer chain used in cellular respiration. Nitazoxanide binds to P4 in a separate domain from pyruvate. However, the binding of the enzyme causes it to release from pyruvate, and when the enzyme cannot contribute to the reaction, it is inhibited. And then no more acetyl coenzyme A, no more electron transfer chain. So, interestingly enough, this parasite is actually resistant to some very common anti-parasitic drugs, such as antifolates, which is a class of drug typically effective against a wide variety of helminthic parasites, protozoan parasites, maybe even other apicomplexans. So it works by inhibiting the interaction between folic acid and methyltransferase. Methyltransferase is responsible for using folic acid to synthesize purines and pyrimidines, in other words, nucleotides to build RNA and DNA. However, Cryptosporidium is known to take these nucleotides from host cells that it inhabits in order to synthesize its own DNA and RNA. Thus, it is slightly resistant now to this class of drugs, and it is increasingly more resistant to this class of drugs as it is used more and more. But for now, it is still partially effective. And here are my sources. Thank you for watching.